Welcome to the screencast of the Big Cat Phylogeny lab. This lab is based on this science article here, the late Miocene radiation of modern Felidae, a genetic assessment carried out by these scientists here, a whole group of scientists. You can read their abstract, the summary that they wrote for their paper about what it is, a lot of complicated words in there, but what we're looking at is the movement of modern Felidae, which is the modern cats, the big cats, around the world 30 to 12 million years ago. So a long time ago, and these movements that they did back and forth. But inside of this paper, what's interesting to us, and this is a 6-page paper, you can see we're on page 1 of 6 here, and it just goes on and on and on. What's of interest to us, we're going to use this paper in order to construct their genetic profile of these cats. They had to obtain actual genes. So what we're going to do, which is the case for many papers published online, they have supplementary materials. So I went to the supporting online material for this paper, and you can see this, they have 353 pages of supporting material. And on page 20 of this, since I've been here before, I can skip ahead, and I marked out here, this is the list of genes that they based their phylogeny of the big cats on. They tracked their big cat movements throughout time on. We're just going to focus on one of these, in which is called the RAG2 gene, the RAG2 gene. Now, the RAG2 gene, what it is, it codes for a protein that's involved in the development of B cells and T cells. You remember those from your our look at the human immune system and the role that they play in your specific defenses. So B cells and T cells, this is a immune system gene that is also present in humans, but it's present in cats. And we're going to use that immune system gene. You can see here that it has 473 base pair alignments, all of which are coding. So we're going to use that RAG2 gene as part of our source for genetic information on cats. Okay, I should point out here that what this means here, it says RAG2 gene DQ082330 to DQ082374. So there's 44 genes there. It's a whole range, so 30, 31, 32, all the way through 74. They have 44 species of cat that they use in their analysis. We're not going to do all 44, but we'll get all those gene sequences. To help us process all the information, we're going to use software that's provided for free. It's called the Molecular Evolutionary Genetics Analysis tool. It's a genetics software, and that's an acronym for MEGA. So the MEGA software program we're going to use. You could download it for free off this website yourself. You can see the web address right there and follow along. It's available on Windows, Mac, and all kinds of other things, Red Hat and Linux if you want to do that. I downloaded the PC version. This is what it looks like. It's just an interface that we can use to get genetic information off of the supercomputer database and analyze it. Okay, so our first step here is to get some genetic information. Inside of MEGA, I click on the Align button here. I'm going to query databanks. Means I'm going to ask a question of a databank. Hitting that opens up a link to a website within MEGA. You can see this isn't the Chrome browser. This is the MEGA web browser. I'm inside that other program. But it's just opened up this website, the NCBI, the National Center for Biotechnology Information. And I can type in any gene information I know into this search bar. What I'm going to do is put one of those gene numbers in for that RAG2 gene, DQ082342, and hit enter. And it comes up with this, right? Puma concolor, that's the scientific name. And it tells me here, Puma concolor, the source, Puma concolor, the puma. So the common name of this is the puma, and it's also known as the cougar, I went to Washington State University, the Cougars. So this is a puma concolor gene. This website has all information about the gene, including the entire nucleotide sequence, all 400 plus nucleotides, the A's, G's, T's, and C's of that gene. What I can do with the MEGA program is rather than copy and paste those things, I can just click on the plus to add them to my alignment. I can give a name to this. Like, what do I want to call my information? But it's just going to use the genus name Puma concolor. I hit OK, and there it is. It opens up this Alignment Explorer, and it's just copied all those C's, T's, A's, G's in here and labeled them. This is the Puma concolor RAG2 gene, and that's the whole sequence there. And if I scroll across, you can see all the colors flash. It's all of those nucleotides inside there. Okay, so I've captured one gene. I need to go in here and capture another. And if I click on the right one here, there we go. I just go back and I can hit the back arrow in my nucleotide sequence and then just type in another one, DQ082343. Hit the enter button and it pulls up all the information about this gene. Now this one is in a Jaguarundi. This common name is a Jaguarundi, Puma jaguarundi, a different kind of cat. If you don't know what that is, you could always type that name in there, Puma jaguarundi, and see what a Jaguarundi looks like for yourself. All I care about right now is adding that alignment sequence. As soon as I click that and hit OK, now you can see in my Alignment Explorer window, I now have two sequences lined up side by side, the Puma and the Jaguarundi. And if I slide my bar over, since they're color-coded, it makes it easy to see if there are any differences inside here. Like right here, I can see there's a spot where a G has become a T, a slight mutation. All I'm going to do now is just repeat that exact same process and copy a whole bunch of nucleotide sequences from a whole bunch of different cats. I'm going to pause the video because you don't need to see that happen. Okay, you can see here I've continued this. I did 12 different cats going down the line here. You can see all their scientific names, and they are all the same gene. So we're comparing variation within the RAG2 gene across 12 different cats. So this gives us 400 plus different nucleotide character states to base our phylogeny off of. I'm just going to note that this last one here, the Linsang, that's actually not a cat. So you can check out what that is. That's the root of our tree. You can look into that one. Okay, now that I have all of these sequences, I need to align them. You might remember we talked about doing a BLAST before, a basic logic alignment of sequence technique. Well, we're going to do the same thing. So I'm going to go to Edit and select all these sequences. You see how I highlighted them all, just copied them all. Now I'm going to alignment. I'm going to align them by cluster all. That just means I'm going to do 12 different ones at the same time. Rather than a BLAST being just 2 side by side, I'm going to do all 12 together. I'm going to cluster them all together. And I run this, I hit the default settings, hit OK, and the computer now, the Mega software, just lined them all up for me inside there, which is kind of cool. And I go across, it's going to have the maximum lineup, but I can see some of these little differences inside here that do exist with the cat. Okay, so I have all this raw data that's been compared to each other. I need to export that data so I can use it for something else. I'm going to go to Data, Export as a Mega format. It's just a file formatting thing. I'm going to call this Mega Cats. Very fancy name that I just gave it. Hit Save. In title input of the data, it's still going to be, it's called Big Cats. And hit OK. Yes. And it's just saved that file for me. Okay, the next and last step of doing this, now that I have all the DNA lined up, is to go back to the main Mega program and make my phylogeny. So I select phylogeny. We're going to do a type of phylogeny called a maximum parsimony, which we're going to make it as happy as we can. Yes, we're going to use the current data that we have. I want to use a bootstrap test for my test. Hit compute. I'm just going to accept all the defaults, and it plugs away at the computation. And what it's doing is comparing all the possibilities. Like what's the best match, and it's going to make every type of tree that's possible mathematically and test to see which one has the strongest support overall. That's what maximum parsimony means. So which one has the most number of nucleotides matched up within the tree, so the fewest number of mutations to produce all those different cats, because that's the most likely historical scenario. And that's what it's doing. You can see it takes a long time. It's actually computing this as this goes on. This is my computer doing this, so that's why it's taking a little bit of time. If you had a faster computer, it might go faster, slower one goes slower. You can see all the parts of the test here, what it's doing in more detail than we need. I'm going to pause while this runs the test. Okay, it's almost done constructing the tree, so we should shortly see this window go away, and it's going to ask me, oh, it failed with a couple of nucleotides. That's okay. Failing 13 times out of the thousands of computations it did is no big deal. So now we get a phylogeny, but remember, we added one species that was not a cat to be the root of our tree, to be the not cat, identify that. So this tree is not quite done yet. I need to add the root to my tree. There's a little function over here that says place root on branch. I click on that and I go find that non-cat thing, which was the banded Linsang. I click on it, And now that's made the banded lincing. I'm going to click on the arrow so I don't reroute my tree. Now the banded lincing is down here as the outgroup, and everything else, this whole group here, this is the cats. These are the cats inside here. And I have different numbers here. These numbers represent what are called bootstrap values. That's a statistical term, a bootstrap. And all we really care about is numbers that are over 50 are going to be significant for us. That's a major group that this tree is very positive about. So numbers over 50, and I can see there's only 2 of them that are the really big numbers. Here's one that's 60. So here's an ancestral group, with this being the common ancestor, and these 1, 2, 3, 4 cats are tightly linked as a family. And there's another family group over here, the 79 group, and I have 1, 2, 3, 4, 5 cats that are strongly linked inside this family. So I've got one family of 4 and one family of 5, and I can look at these and wonder, what are these cats? So the first thing that I notice when I look at this group of 66 up here, these cats, this one here, Antonix jubatus, that is a different cat than the what jubatus is. If you type in its scientific name into Google, that's a cheetah. Then a cheetah is an African cat. Cheetahs live in Africa. But puma concolor, the Diagarundi, and Leopardus pardalis, type those in, you'll find out these are all American cats, North and South American cats. So this group here of 66, this is a group of New World cats. All these cats live in the Americas. But we have a cheetah here, and that's a little odd, because cheetahs, all living cheetahs, are alive in Africa. Looking at this group of 79 here, this is a big group of cats, and you can see here's Panthera pardalis, Panthera tigris, the tiger, Panthera onca, Panthera leo, that's the lion, and the Uncia uncia. You can look up what these are. The one that sticks out to me is Panthera onca, because that is a jaguar. Jaguar is a New World cat that lives in America. You're familiar with the Jacksonville Jaguars. They're named after the cats that live in that area. But all these other ones, tigers, lions, leopards, these are all Old World cats that live in Africa and Asia. So here we have the jaguar, a New World cat, that is in the family, in the group here, the clade of Old World cats. So we need to explain this phenomenon. That's what's interesting about this family tree. Now I can go up here and I can save this tree, and I will do that. I'll save it as a GIF or some kind of image. I can save this thing as a TIFF or a PDF file, and I can upload that. It's called Big Cats, and then I'll put that up there for you guys to take a look at and to analyze. So that's the video, how to, based off of the initial paper of Johnson et al. On the radiation of modern cats, and we ended up with this whole phylogeny of the cats based on one gene. Now, had we used all the genes that they had in their paper, and remember they had many, we'd get an even stronger tree, but this is enough for us to see how that kind of genetic analysis is done using all of these base pairs as our character states, which is kind of cool. Okay, thank you.